Now today we want to discuss a new chapter. New chapter, not new chapter, new topic. Artificial hybridization. Artificial hybridization. Now, before discussing this topic, what is the natural phenomena to this title or topic? What is the topic? Artificial hybridization. Artificial indicate man-made. We are conducting. It is not a natural. But what is pollination is a natural. Right. Why we are going to the artificial hybridization? Why we are not interested in natural? Why we are entering into the artificial hybridization? That means, right, to increase the yield in the crop improvement. That is the main aim, significance of artificial hybridization, importance of artificial hybridization for crop improvement. And it is the main technique, in plant breeding. Plant breeding is a separate branch of biology. Right. A separate chapter is there. In future chapters, what is plant breeding? We can discuss later. This separate chapter is there. Right. Now, what is the topic? Artificial hybridization. What is the need? What is the definition? Now we are going to the discussion. But before this, why we are conducting artificial hybridization? Crop improvement. Crop. Plants which grows in forest, wild plants. But some plants used in agriculture, the plants which are used in agriculture are called as crop. Due to increasing of human population, right, the yield is not sufficient. Due to this, right, due to scientific knowledge, we can improve the yield of crop. Right. For the improvement of crop yield, yes, this technique. Of course, natural pollination will occur, some yield will be there. But that yield is not enough to growing population. Due to this, some scientists works on this technique and given the farmers, right, these farmers are using this artificial hybridization and day by day increase the crops. This is the main concept of artificial hybridization. Right. Now, it is a main technique in plant breeding. Right. Now, what is the definition of artificial hybridization? A crossing between two plants. The two plants may be same species, or different species. Is it? Right. Now, the crossing between two plants differing from each other genetically in our desired character. Such cross is called artificial cross. Now, two important steps. Yes, emasculation, bagging. This is the two steps involved in artificial hybridization. Now, what is this emasculation? Right. Now, suppose it is a sugarcane plant. It is also sugarcane plant. Sugarcane. The scientific name of sugarcane, Saccharum officinarum. Okay. Now, suppose it is in your local area, sugarcane, local area. It is the long stem. Internodes are long. Now, this is the node. It is the node. The length of nodes is very round, but thin. Now it is another area. Right. Now see. Now thickness is more, length is short. But any agriculture farmer, they want the length node is more and thickness is also. Now that is our desirable character. Length of node, that means length of stem. Length of stem is more and thickness is also more. So the man is always think commercially, with less time, more benefit. Right. Now, it is a sugar cane in one area. That may be North India or South India, whatever it may be, one area. Right. Now, what is the morphological character? Right. Node. The node is long. Long. But thin. Thin. Here the node is short but thick. Round. When you cross short node with thick is crossed with long node thin. Then in the next generation you may get, but not 100% guarantee, but you may get long node thickness. So desirable character is there. For this, right, a artificial cross is there. Now, see, Now, it is a sugar plant, it is a sugar plant, or mango, whatever it may be. So, two plants. In these two plants, you have desirable character. Now, you know, pedicel, thalamus, sepal, flower, as anther, stamen, now, pistil. Now, it is another plant, right. Right. This is the two desirable plants selected for artificial cross. Now this plant is containing flower. The flower is a bisexual. It is also bisexual, it is also bisexual. Okay, you think, right, you are taking pollen grain from this plant or from this plant. Just before that, you may decide, right, if you take the pollen grain from this, it becomes male plant. Is it? But basically it is a bisexual. It is also bisexual. These two plants is having our desirable character. Due to this, you are ready to cross it. In this crossing, right, you collect the pollen grain from one plant and keep on the stigma. Suppose you collect the pollen grain from this plant. That means it is the male. It is the male. Right. Choice you may consider it is a male, whatever it may be. Correct. Male means taking of pollen grains. Now see. Right. Now it is the male. Now it is the female. Right. Now in this female plant, in bud condition, with the help of scissors and forceps, you remove the stem in bud condition. Right. Now it become unisexual or bisexual? Unisexual. Staminate or pistil? Pistil. Because stamen is removed. Now a process conversion of bisexual flower into unisexual as a pistillate is called as emasculation. Once again. Now, right. Now, it is the pedicel, thalamus, petal, sepal. Now, stamens, pistil. Now, this flower, according to your opinion, this plant, in this plant, you are taking the pistil or pollen grain. Pistil. You are collecting the pollen grain in other plant. Right. Hence, based on the pollen grain source, selection of pollen, it is the male, but both are bisexual. But now, in this bisexual flower, as you remove the stamen completely at bud condition. During bud condition, you remove the stamen. Now, by this result, now this bisexual become unisexual pistillate. Now, what is this first step? Emasculation. Emasculation is occurred. What is emasculation? A process conversion of bisexual flower into unisexual as pistillate. This process is called as emasculation. Immediate after emasculation, right, you take some cover which is made with butter and transparent material and cover the total flower. Right, after emasculation, you cover the total flower with a transparent bag. This is called as bag. What is the need of bagging if you are not covered with any transparent bag? Now, unwanted, undesirable pollen grain will be deposited. To prevent the deposition of unwanted, undesirable pollen grain, right? You cover the bag. This step is called as bag. Now, this is the taken in male plant or female plant? Female plant. Now it is the male plant. How can you say it is a male? Because you are taking pollen grain. On this basis, it is the male, but actually it is a bisexual flower. Now when anther is dehiscent, right? This dehiscent anther take, right, with manual, right, and dusted, keep on this pollen grains on the stigma. Here, there is no natural pollination. The pollination by a farmer or a scientist. Hence, it is artificial hybridization. So, this pollen grain which is dusted, which is deposited on the stigma with our desirable plant. It is our desirable plant. From this our desirable plant, right, take the dehiscent anther and keep on stigma. Again, then you rebagging. Again, it prevents the unwanted deposition of unwanted pollen. Now, this is the two important steps. But sometimes, if you take the papaya, basically the flower is female, right? If the flower is female, unisexual female, there is no need of emasculation. Emasculation is necessary for the conversion of bisexual flower into pistillate. But basically it is a pistillate, but there is no need emasculation. At that time, only bagging is necessary. Is it? Now this is about the artificial hybridization. Now come to the next topic, double fertilization. Now another new topic, double fertilization. It is the most important examination point of view, and it is also interesting topic. Generally, if any organism origin of any organism, in the origin of man, in the origin of frog, right, only one fertilization is there. So in my origin, fertilization is there. What is the fertilization? Right. It is the sperm. It is the egg. The union between male and female haploid gamete to form diploid gel. This is called as fertilization. The fertilization is the most important event in all sexual reproduction of organism. Without fertilization, there is no sexual reproduction. What is the chapter name? Sexual reproduction in flowering plants. That means fertilization is the major event. But when compared with angiosperms, in other organisms, bryophytes, animals, they require only one fertilization, but they shows two types of fertilization. Hence, it is called as double fertilization. So, in the sexual reproduction of angiosperm, in the sexual reproduction of angiosperms, two types of fertilization. Right. Now, definition. In the embryo sac of angiosperm, two types of fertilization. That is, fertilization means fusion, not fission. Fusion. That means togethering. Alright. Now, two types of fertilization, that is fusion, occur. How many fertilization? Two types. Among these two types, syngamy is the name of one fertilization. Triple fission is another name of other fertilization. Catch the point. How many fertilizations? Two fertilizations. Right. In these two fertilization, you take any fertilization. Fertilization means union. That means fission. Right. Among these two fertilization, name. First two fertilization is called syngamy. What is the syngamy? According to this title, it is one type of fertilization. What is another type? Triple fission. Hence, the fertilization of angiosperm is called as double fertilization. It is the unique in angiosperm. We can see double fertilization in only angiosperms. Other than angiosperms, you go to the animal or plants, whatever it may be, they require only single fertilization. This is the difference between double fertilization and single fertilization. Now, in detail. Right. Now, how many fertilizations? Two types. Among these two types, what is this one? Single. Now, see. Right. In two types, one is syngamy. The another one is triple fission. Right. These two types of fertilization in angiosperm, where it is occur, what is the site, what is the place? Embryo sac. Hence, I given the in embryo sac. In the definition already I given the in the embryo sac of angiosperms, two types of fertilization occur, namely singamy and triple fission. Hence, the fertilization of angiosperm is called as double fertilization. Right. Now, it is a embryo sac. Now, before embryo sac, just listen. Right. Now, right. What is this total? You know, pistil. Among this pistil, now we take this one only, ovary. Inside the ovary, ovule. Inside the ovule, embryo sac. Inside the embryo sac, egg. Egg. Now I take embryo sac. Now, what is the level of embryo sac? What is the level of embryo sac? 7-celled, 8 nuclei. Now, 1, 2, 3. These are antipodas. How many antipodas? 3. 1, 2, 3. This is 2 are synergities egg. Altogether is called as egg apparatus. Egg apparatus. In egg apparatus, this two is called as synergities. How can you identify the synergy? Some thickening filiform apparatus. Now it is called as egg. Now, these three cells, total 6 cells. This 6 cells is accommodated in this cell. This is LCC, large central cell. Now, what is this? Cytoplasm. Now, what is this? Nuclei. This nucleus is the 2 nuclei, binucleated. This binucleated to whom? Central cell. That means in the cytoplasm of LCC, 6 cells are accommodated. These 6 cells plus large central cell, 7. But large central cell containing how many nuclei? 2. These two nuclei of large central cell is called as polar nuclei. Now this is about the embryo sac. Now, before going to the double fertilization, you must idea what is the structure of embryo sac. Right. Now, see. Now, I am going to draw the embryo sac once again. Now, it is embryo sac. Is it? Now what is this? Cytoplasm. Now, plasma membrane and cell wall is also there. Cell wall. Right. Now, what is this? 2 polar nuclei. On one end, a set of 3 cells. This is called as antipodas. Another end is a set of 3. 1. Right. Piliform apparatus. Now it is called as egg. Altogether is called as egg apparatus. Oh, now this is okay. Now, just a minute. Pollen tube. Now it is a pollen tube. Right. Which is coming from stigma. ovary, embryo sac, pollen grain. Now what is this? Pollen tube. The pollen tube gradually grows and penetrating into the tissue. Now, right. This is the pollen tube. This pollen tube I draw here. Right. Now which containing two male gametes. Two male gametes. Right. Now, see. That, right. This pollen tube, tip of the pollen tube touch the synergetics. Filiform apparatus guide the pollen tube to penetrating in synergetics. Due to this, right, these two male gametes which is carried by pollen tube is enters in synergetics. In only one synergetics. Is it? Now what is this two? Male gametes. Right. The male gametes is present in where? Synergetics. Among these two male gametes, one male gamete is fused with egg and form zygote. This is called as syngamy. The syngamy is the formation of zygote by the fusion of one male gamete with egg. This process is called as syngamy. Now, what is the remaining other male gamete? This other male gamete moves toward the center, right, and fuse with two polar nuclei. One. What is the name? Polar nuclei. What is the name? Polar nuclei. These two polar nuclei, and it is 3. 1, 2, 3. Fuse and form nuclei, not cell. Nuclei. That is called as pen. Now, the pen is the result of fusion of two polar nuclei plus one male gamete. That means here how many fused? One male gamete and two are polar. That is how many fused? 3. Hence, it is called as triple fusion. Triple. What is the meaning of triple? 3. Who are 3? 2 are polar nuclei and one is male gamete. These three fuse and form pen. Right. Which process involved for pen formation? That is called as triple fusion. Right? Now, what is the next future? Now, before going to this, I forget one thing. Just listen. Right. Now, it is embryo sac. Right. In this embryo sac, it is LCC, large central cell. Now what is this? Cytoplasm. Now what is this two? Polar nuclei. What is this 3? Antinucleus. Now name these two cells synergities. What is the future of the synergities? Filiform apparatus. Now what is this? Now, what I am forgetting, just now I am going to explain. Alright. 1, 2, 3, 4, 5, 6. The 6 cells accommodated in LCC all together is called as embryo sac. So, LCC is a part of embryo sac or embryo sac? Part of. All together is called as embryo sac. In embryo sac, LCC is one part. In embryo sac, antipodal is one part. In embryo side, egg is a one part. Okay. What is this? Egg. Plied. Haploid or diploid? Haploid. Synergities. Haploids. One set. This is haploid, this is haploid. What is this? Polar nuclei is also haploid. It is also haploid. So, total in embryo side is haploid cells. 7 cells is there. Among the 7 cells, each cell is haploid. Okay. Now, see here. Just 2 minutes. This whole right now. It is not. Right. Antipodals. Polar nuclei. Right. Now what is this? Pollen. Now it's containing male gametes. How many male gametes? 2. Plied, haploid. It is haploid. Haploid is represented by N. It is also N. Now what is this? N. What is the plied? N. Now what is this? Thickening filiform apparatus. Due to presence of filiform apparatus, now synergity is attracting pollen. Right? The filiform apparatus guide the pollen tube to entry in synergities. Now, the tube will be dissolved. 2 male gametes is released in synergities. In only one synergities. Now, what is this male gamete? This male gamete is fused with N and form zygote. What is the plied of zygote? Diploid. How can you say diploid? Because it is formed by 1 male gamete and 1 egg. Male gamete is 1 set, egg is also 1 set. Hence it is a zygote. Now zygote is diploid. Okay. What is another one male gamete? Right. Another male gamete is moves towards center. Now it is the male gamete. What is the plied? Haploid. What is these 2? Polar nuclei. Each polar nuclei is haploid. Haploid. Just listen. Haploid. How many haploids? 3. These 3 haploids fuse and form nucleus. That is pen. Now you estimate what is the pleiady of pen. Haploid or diploid? No, neither haploid nor diploid. It is diploid. Because now it is a pen. Just before formation of pen, right? 2 polar nuclei and 1 male gamete. Male gamete is haploid. It is haploid. How many haploids? 3 haploids. 3 haploids together form triploid. Is it? Now, after this, after fertilization, right? Now, see here. I wrote the 2 scientists' names, Strasburger, Nowshad. Strasburger is given the definition of fertilization. Fertilization is first introduced by Strasburger. Right. Be careful. Double fertilization is the first noticed by Nowshad. See, fertilization is a notice. Normal fertilization was noticed by Strasburger. That is a fusion of male and female gametes to form the diploid zygote, which is a common in majority of sexual reproducing organisms. That is called as fertilization. Right. But in angiosperms, double fertilization is occurred. That is noticed by Nowshad. Now, after the fertilization, right? After the fertilization, double fertilization. Now, right? What is present in the center after fertilization? Pen. Primary endosperm nucleus. Primary endosperm nucleus. Right. What is this? Zygote. Right. After the double fertilization, what is these three? Antipodes. They are degenerating. This degeneration is represented as this line. Dots. Now it is degenerated antipodes. Now what is this two? Synergitis is also degenerated. Now, see. Now, it is now fertilized embryo sac. How can you say it is fertilized? There is no antipodes, there is no synergitis. Right? Now, instead of polar nuclei, pen is there. Instead of egg is zygote is there. Now this is called as fertilized embryo cell. Now this pen, plied pen. Now this pen, syngamic result, zygote, triple fission result, now the zygote, right, in the next step become embryo. When you are mother womb, embryo. The embryo formed from zygote is formed by the fertilization. Similarly, what is this? Embryo. Embryo is developed from zygote. For the formation of zygote, this process is occurred. Without syngamy, there is no zygote. Without zygote, there is no embryo. Without embryo, there is no next generation. Now, what is this? Triple fission. Now, it forms pen. Isn't it? It is in the next step. It forms endosperm. Right. For the formation of endosperm, pen is required. For the formation of pen, this event is occurred. Name the event. Triple fertilization. Triple fertilization, right. Result is pen. The pen is undergoes division and form endosperm. What is the endosperm? Is a nutritive tissue for growing embryo. When you are in mother womb, some placenta will be there. This placenta supply the food for growing embryo. Similarly, this embryo requires some nutritive tissue. That nutritive tissue is nothing, endosperm. So, this endosperm is a tissue. It is a pen, nucleus, it undergoes division and forms tissue, triploid tissue. Like endosperm is a triploid tissue, act as nourishment for growing embryo. This is the definition of endosperm. Finally, I am closing. Now, what is this? Embryo. This embryo requires, right? This embryo formation requires zygote. Zygote formation requires syngamy. Right? What is this? Endosperm. Formation of endosperm from pen. Pen is not a cell, just it is a nucleus. This formation of nucleus requires triple fission. But what is the significance? What is the need of endosperm? Right. Nutritive tissue. Triploid tissue which is act as a nourishment for embryo is called as endosperm.